In examining the rationale for FDA approval of the 21 drugs with at least one null primary endpoint, we found that the most common reason cited by the FDA for approval was the drug success in at least one other pivotal study. The next most common reasons were favorable post hoc analyses or positive findings from secondary or exploratory endpoints in the pivotal studies. So here's an example of one of the 21 drugs that had a null primary endpoint. This is for a combination antibiotic of celastin, imipenem, and relebactam that's marketed as Rocarbrio. And it's been approved by the FDA for the treatment of complicated UTI as well as complicated intra-abdominal infections. There were two pivotal clinical trials that supported these indication approvals, and they had two primary endpoints. And the endpoints were also assessed using a non-inferiority hypothesis. The one endpoint that was found to be null was a primary composite endpoint of combined clinical and microbiological response for complicated UTIs. And instead, FDA actually based its approval on the success of in vitro and animal studies, as well as historical data demonstrating efficacy of one of the drug components, just the imipenem portion. In fact, they actually did not consider either pivotal study for complicated UTI and complicated intra-abdominal infection as a part of their approval rationale, because they found that the trials were actually not adequately designed for non-inferiority testing. So it was just interesting to see that despite the primary endpoint not being missed, but also the trials being not adequately powered or designed for hypothesis testing, FDA disregarded that, instead used other supporting data and not really trial data for their approval decision. So just for conclusions, 10% of the drugs that were approved by FDA between 2018 and 2021 were supported by pivotal studies with null findings for at least one primary efficacy endpoint. The most common rationales were meeting primary endpoints in other pivotal studies, either positive or exploratory endpoints in pivotal studies, and then favorable post hoc analyses. And about 1/3 of the drugs with null endpoints had post-marketing requirements addressing these null endpoints. So the implications of this, it underscores really the complexity of regulatory decision-making that FDA has in making these decisions. And it also points to the need for further post-marketing requirements and their timely completion to address areas of clinical uncertainty, especially when these endpoints are not being met, and to also inform clinical decision-making. And also highlights also the need for improved communication to patients and clinicians on the basis of FDA approval in the setting of null primary endpoints. We had to dig through approval packages, looking at the different endpoints to be able to determine which were missed and which weren't. And these are not, clearly indicated on drug labels necessarily. And so improved communication even through that as a first step would be helpful, both in the clinical setting, but also, for further scientific review of these drugs. Our limitation, this was focused for trials that were submitted to the FDA, despite FDA possibly considering other studies or even other regulators' decisions in their approval decision-making process. And then moreover, withdrawn or rejected new drug and biologic applications were not analyzed, as that information is not public. So I just want to say a special thank you to James Johnston, former CERSI scholar, who I think is on this call, who led the analysis for the study, and then also Joe Ross as well.
Thank you so much. This is really fascinating. Can you talk a little bit about whether there were any of these drugs that were far enough along that you were able to look at post-marketing studies, or whether that comes out any different, or is that like a next step in the study?
Yeah, that's a great question. We only look to see whether or not the post-marketing studies address the primary endpoint. So among the 7, that would be a great next step, to actually look at the 7 drugs where they do have post-marketing requirements addressing the missed endpoint, to see if there was one, did the endpoint continue to be missed? And would that then change clinical decision making, or if they have been met, and then that supporting FDA approval. But yeah, that would be a great next step.
Yeah. The question in the chat, sometimes the failure of the endpoint is due to the hierarchical order of the endpoints or use of statistical power for an early look. So really due to statistical uncertainty. So since they're so expensive and that's a limited resource, do you have any recommendations from this to overcome those hurdles? Did you look at the extent to which any of these were null for that reason?
So we did, and we have that, we detailed like all 21 of them in our as a part of the analysis. And so there were, yes, a couple of them for sure where that was the case, especially for some drugs that were indicated for very rare diseases or for neglected tropical diseases. There was a couple of them in which, that was an issue. But I think this kind of like points, again, yes, the studies are expensive, but the importance of FDA having kind of oversight on post-marketing requirements and continued surveillance as well, to kind of see if it's not going to happen, approval stages, and they're making these approval decisions. And there is, kind of maybe perhaps potential for these drugs to be effective regardless of the missed endpoint, then, that we need to have post-marketing requirements that do address this. And so that would hopefully, raise the sample, increase power so that we can be able to answer these questions more definitively.
Yeah. And I'm so sorry, I don't know your background. Are you a physician?
Yes, I am.
Okay, so tell me, I'm really curious about your suggestion about communicating this to clinicians and what the FDA can kind of do for that. I was just talking to some clinicians about the labeling of opioids, and they were like, labeling, like they weren't really quite sure even like what I meant exactly by that. So can you talk about that?
Yeah, in clinical practice, that's a really great point, because in clinical practice, I wouldn't say like the typical physician, we don't pull up drug labels to exactly look at them. It's oftentimes through guidance, but those drug labels do inform like clinical practice guidelines, and and so that piece, I think that translation, if you're starting with the labels even as a first step, that then informs guidelines, because those are oftentimes used as the basis for making those types of decisions, maybe not going to in-depth in terms of looking at the medical review. That might be at least a good first step. But FDA does have, during the pandemic, I think we saw FDA communicate in many creative ways directly to patients and clinicians around COVID-19, therapeutics and vaccines, and adopting some of those lessons learned might also be appropriate in these cases, especially when there might be uncertainty in which there might, concerns around risks outweighing the benefits come into play, and where FDA has continued questions that they are asking post-marketing requirements to be addressing those issues.
Great, thank you. So we have 3 minutes. It looks like most of the questions in here were kind of responded to.
Molly, maybe Ryan could address your answer directly, because I think there's a lot in there.
Okay, yeah. So I asked Brian whether this data exclusivity is really interesting to me, whether US market exclusivity allows those other countries to rely on data exclusivity.
So I think in part the US market exclusivity or the US regulatory exclusivities provided by FDA can help the other countries rely on data exclusivity or less. But generally, I think it's more the patent landscape that has the stronger role in preventing the biosimilar entry. Usually the patents last longer than the regulatory exclusivities provided by FDA. And in particular, there's already been experiences with in Humira in particular, the adalimumab biosimilars in biosimilar patent litigation in the US, where prospective entrants were trying to come in earlier. The originator negotiated allowing them to enter the EU market earlier. So by taking advantage of the patent and regulatory landscape in the US, they can use other jurisdictions as a bargaining chip. So them having a slightly lower or less exclusivity will ultimately benefit other jurisdictions because they'll have earlier biosimilar entry. And a lot of originator companies will feel comfortable, given the very profitable US market, they have a lot more security. And there's been other evidence supporting that certain patents aren't filed in other jurisdictions, but are filed in the US. There's more litigation in the US versus in EU and any of the other jurisdictions. So that's another big area we're going to dive into more the next project, or the next phase of this project.
That is fascinating. Well, thank you all so much. This was a really terrific panel. I really enjoyed all of these presentations, and I'm looking forward to seeing the final papers for them. Nice work, everyone.
Yeah, this was really terrific. So I love the chat that's going too, so please keep it up. Molly, I'm just going to say out loud, my internet cut out in the middle of the last one, so if you have the whole chat, can you try to cut and paste as best you can?
Yes, I will do.
Thank you. Yep. Because Tina Morrison asked that we hold it, because I know she had to drop off.
Yep.
So our first session, we tried to organize these talks. It's imperfect because we had no idea what was going to come in. The first one was a little bit about innovation, particularly on the drug and biosimilar side. It wasn't exactly for every presentation, but it lined up pretty well. The next session is kind of like our COVID we session, although Asmita doesn't fit exactly perfectly, but that's okay. So we have 4 good presentations that we're going to kick right into, and we are going to start. And again, just as a reminder, everyone introduce yourselves and kind of where you're at in your career and what training you're in when you start your presentation. But our first one for the hour is from Usman Manir, who's going to be talking about the agreement of treatment effects from observational studies and clinical trials evaluating therapeutics for COVID. So Usman, share your screen and take it over. Do we have Usman? I didn't confirm that he was on.
Sorry, my mic was muted.
Oh, there we go.
Let me just share my screen and make sure it's in presenter view. Okay, can you see this?
Yes.
Okay. All right, great. So hi everyone, I'm Usman. I'm a medical student here at Yale and part of the Yale CERSI program. Today I want to talk about the agreement of treatment effects between randomized control trials and observational studies for a few candidate therapies for COVID-19. We conducted the study as a meta-epidemiological study, and wanted to acknowledge Dr. Ross and Dr. Wallach for their excellent mentorship along the way. All right, so starting things off, for a bit of background on the question at hand. Randomized controlled trials, RCTs, as everyone probably knows at this point, are considered to be the gold standard generally for determining the safety and efficacy of treatments. They're generally what's used to determine drug approval, and we rely on them pretty heavily both in the clinical and regulatory settings. But they have their own drawbacks, namely limited generalizability and also requiring a significant amount of time and resources to plan. COVID, in the beginning of COVID especially, highlighted some of these shortcomings, and there was a potential tease for this role of observational studies, these studies that are conducted through non-interventional methods by reviewing patient records and by reviewing already existing drugs, to potentially look into the clinical value of candidate therapies that were already approved and already on the market that we had data for. But there were concerns about the low quality of the evidence and the potential quick dissemination of evidence that might not be up to standard. So little was known about the agreement between RCTs and observational studies, specifically whether or not those treatment effects are concordant, whether they agree with each other. And so that's a question we sought to ask. We wanted to see whether or not there was agreement between treatment effects from individual or meta-analyzed observational studies and corresponding RCTs that looked at the exact same clinical question, which we defined as the same comparator and outcome for of COVID. This question, we went through a very systematic methodological process. I just highlighted the overview of some of the steps along the way, and I'll dig deeper into a few of these. So we first identified which interventions to actually use systematically. We then identified the individual observational studies and the individual RCTs to consider. From there, we matched the observational studies and RCTs so that studies were matched according to the same corresponding clinical question. We extracted data and analyzed the data by first meta-analyzing the observational studies into groups of the same clinical question. Subsequently, looked at the summary demographics across these pairs of observational studies and RCTs. From there, we compared the treatment effects. That's where the money's at in the study, of course. And then we compared the publication timing across these studies, conducted risk of bias assessments, and did some sensitivity analyses. Digging deeper into the actual methods for the identification of the RCTs and observational studies, I wanted to highlight that we didn't want to reinvent the wheel for identifying RCTs. And so we relied on a meta-analysis, living review of randomized controlled trials for several COVID therapies that was published in the BMJ. And then we identified observational studies by leveraging an evidence database that essentially did the PubMed search of looking into indexing interventions, comparisons for COVID-19. In a quick overview, we started with a large number of records, around 5,000, and then we screened around 3,000 of them and ultimately included 46 observational studies in our paper. Spending some time on the data extraction and analysis component, I want to point out that we pulled from both the observational studies and RCTs, besides information about the study demographics, the study design components, also information about the outcomes, and that we grouped into two main categories: the dichotomous outcomes that we looked for odds ratios, and continuous outcomes that we looked for mean differences or ratio means. And when it was possible, we reanalyzed the outcomes from the individual studies and meta-analyzed them. And we also reanalyzed the data from the RCTs included in the living review that we leverage for our study. I also want to spend some time talking about how we define concordance of treatment effects, because this is really crucial to our study. The whole point is that we wanted to identify how often these studies agree in their treatment effect estimates. And so we defined concordance in three ways, essentially. On the right, you can sort of see this diagram that I sketched out. The first is if the matching pair of RCTs and observational studies are not significant at all. And so that would be a concordant result. The second is if both the RCT and observational study were significant and agreed in the same direction of the effect estimate. That is 2 and 3, or they increase both or they decrease both. And so those are the three schemas we use to define concordance. Turning to the results, starting with the demographic and study results. Table 1 on the side, you can see shows an overview here of study design, masking, and then also demographics. I'll point out that we noticed that in our sample, we included 46 observational studies and 37 and matched observational studies and RCTs have the same median number of included studies. That is when considering studies in a matched pair. They also had, for matched observational studies, a similar but lower number of total participants. And when considering demographic information about age, sex, and disease severity for the patients in the respective studies, we found that they were concordant across all three domains. So that was good to see and reassured us that the studies we're looking at were actually somewhat similar in terms of the composite of population makeup. Turning to the results on concordance for the treatment effects, here I'll show the table that depicts whether or not each individual study in a matched pair was concordant. Overall, of the 27 matched pairs of RCTs and observational studies that we had assembled, we found that a significant majority of them, 21 of the 27, were in agreement. When breaking this out based on whether or not the studies had pooled evidence, or sorry, whether the matched pairs had pooled evidence or whether they had individual studies only, we found that when pooling evidence across studies, that is within a matched pair, if there were multiple observational studies that were meta-analyzed or multiple RCTs that were meta-analyzed, we found that those meta-analyses, when compared together, were typically more concordant than the case in which an observational study representing a clinical RCT was one finding we saw when subsegmenting the sample. And second, when we compared outcome type, when we looked at whether or not a study or a clinical question was looking at a dichotomous outcome, something like mortality or hospitalization, compared to a continuous outcome, something like time to clinical improvement, we saw that the dichotomous outcomes also had more, also had greater agreement. And you can see the percentages as shown. So I wanted to highlight a few large scale themes that emerged from our study. One, it's that across all the matched observational study and RCT pairs that we considered comparing 3 interventions for COVID, we found that more than 3/4 had treatment effects that were in agreement. 2, overall, the agreement was higher in matched pairs of meta-analyses of studies rather than those with single studies. and in those evaluating treatment effects for dichotomous outcomes rather than continuous outcomes. And three, despite concerns that I alluded to in the beginning about individual observational studies, about evidence from individual observational studies evaluating COVID, meta-analyzed evidence from observational studies can complement but may not replace RCTs for determining the clinical efficacy or safety of candidate treatments. So overall, we found that in the future, for potentially quick decision-making in future pandemic settings, accumulated evidence from observational studies could complement RCTs in determining the clinical value of a therapeutic. A few limitations in our study that I wanted to highlight. One is the number of interventions. We only looked at 3 interventions for COVID that were evaluated. We focused on the 3 most important ones, but we did not look at the entire field, of course. 2, the number of eligible studies may have been limited given the approach we used to leverage existing. 3, the timing of publication. I didn't show this on our slide for the interest of time, but we did find that publication timing was, observational studies were typically published before RCTs, but it is still unclear whether one study design prompted another. 4, study characteristics. We matched them according to demographic, but this, of course, is not perfect. And 5, we didn't have access to the raw data, but we did what we could based on the published and preprint data available. So I'll pause there at the 10-minute mark and open for any questions or respond in the chat as needed. But thanks for your time.
Usman, that was terrific. And you're not boasting enough that this paper's been accepted for publication in the BMJ, which is why all your tables and figures look so good.
That's right. The technical editors helped out, of course.
There's some great questions in the chat, but I'm going to save them for the Q&A after because I think it'll lead to good discussion. Let's go to our next presentation from Vishal Shah, who's going to be talking about laboratory correlates of infectivity for SARS-CoV-2. Just a reminder to introduce yourself.
Sorry, can you see my screen here?
Yes, great.
Oh, great. Hi, my name is Vishal Shah. I'm an internist and currently a research fellow in the Evidence-Based Practice Center at Mayo Clinic. Today I'll be presenting on our project called Laboratory Correlates of Infectivity in SARS-CoV-2. Unfortunately, we ran into some logistical challenges over the past few months and are just now getting underway, so I don't have data, but I'll share the background and methods we'll use and along with some implications and limitations of this data. Oh, all right. So each of these topics in the background can be full-length talks themselves, but for the sake of brevity, I'll provide a general overview. We know from earlier data that individuals may be infectious for up to 10 days, based largely on data showing limited ability to grow live virus beyond this time. Additionally, data from large contact tracing studies indicate limited transmission after 6 days from symptom onset. However, the duration of transmissibility may be longer in certain hosts due to immunocompromised state or in those with severe disease. And most of these studies occurred prior to the introduction of vaccines, having large proportions of the population with prior infection and the emergence of new variants. Additionally, and importantly, the duration of transmissibility has largely been based on time from symptom or test onset, though different testing strategies may also be useful. And we'll discuss some of the available tests we have for SARS-CoV-2. Our most prominent, most well-known is the PCR polymerase chain reaction. It's used to amplify and detect a portion of the SARS-CoV-2 genome. It may remain positive for months after initial infection. Typically, it's a binary outcome, either positive, negative, but may be semi-quantitative using the CT value. The CT value is short for cycle threshold value, is the number of cycles needed to replicate to a threshold number of copies of the genome. This may be different based on the sample source and the equipment used, so it's hard to compare across different tests. And it's correlated to viral load. There's some important limitations on this correlation. And some other tests that we have, viral load can actually be determined through ddPCR, digital droplet PCR. And studies have shown absence of transmission of SARS-CoV-2 at viral loads less than 4 log 10 copies per ml. Additionally, we have antigen tests, and smaller studies have shown a correlation between antigen positivity and viral culture. And of course, finally, we also have viral culture, though Requirements for a BSL-3 laboratory and technical challenges limits its use. However, it provides useful information on the presence of replication-competent viruses. So with this background in mind, we wanted to know, is there a test or a combination or series of tests that can correlate with communicability in an individual newly diagnosed with the SARS-CoV-2 infection? So we're testing this, we're testing this research question at Mayo Clinic in Rochester with healthcare worker participants, a vast majority of whom are vaccinated. And the study will have a combination of healthcare workers with direct and non-direct patient care responsibilities. And so we'll identify and enroll up to 100 newly diagnosed healthcare workers through our occupational health services. We'll send a baseline questionnaire to identify symptom onset, prior infections, vaccination history, and any conditions or medications that may impact the immune system. These index cases will have additional testing performed at 2 time points between days 5 and 10 of symptom onset, separated by at least 48 hours. And so one will be a nasopharyngeal sample collection, and we'll run a couple tests on the sample, RT-PCR, which will be used to determine the CT value, the quantitative PCR, and a viral culture. And we'll ask participants to also perform a home rapid antigen test. Again, this will be at 2 time points. In addition to enrolling these COVID positive index cases, we will also enroll their household members. We won't be testing household members, but instead we'll be sending questionnaires at 2 time points at the time of enrollment and around 10 to 14 days. And this questionnaire will identify time spent with the index case, symptoms, symptom onset, their prior infections and vaccination history, and any conditions that can impact the immune system. We'll also check their medical review for SARS-CoV-2 test results. Because of funding, we are not able to test them ourselves, but we will encourage them to follow current guidance on testing. These are our inclusion and exclusion criteria. We're including healthcare workers in Rochester, Minnesota. We are excluding hospitalized workers at the time of diagnosis and those who are not able to perform testing. For household contacts, we just ask that they are living with the index case, and we're excluding them for that episode during the time of enrollment. And our outcomes, we're looking at, our primary outcome is really the proportion of healthcare workers with viral culture positivity at days 5 to 10, with secondary outcome of proportion of healthcare workers with transmission to household contacts. And we'll plan to run a logistic regression model to assess association between viral cultural positivity and the different SARS-CoV-2 testing, accounting for immunization status, prior infection, any SARS-CoV-2 therapies they receive in immunocompromised state, and age and gender. There's a few implications of potential implications of this data. Having tests that can assess transmissibility of infected persons can allow for greater safety in occupational settings, particularly among critical workforce during surges in infection, for example, healthcare workers, public safety officers, and workers maintaining critical infrastructure. Additionally, an improved understanding of transmissibility in a highly vaccinated population can lead to more nuanced guidance for the duration of isolation. So there's some important limitations to this study. First, evaluating household transmission can be challenging. There's other potential sources of infection, and direction of transmission may pose challenges to evaluation. So we'll take steps to try to get a good understanding through our symptom survey. And we're not able to perform viral culture in every sample due to logistical challenges of sample collection and laboratory capacity. An important note that inability to grow live virus on the viral culture does not preclude ability to transmit live virus. Viral culture is difficult, so false negatives are certainly of concern. And this is a pilot study. We're limited in our sample size to account for potential confounders that may impact our results, such as booster vaccination or prior infection. And a few global limitations. The ability to transmit virus is multifactorial with host and environment specific factors. And the behavior of the SARS-CoV-2 virus in the host may change with new variants, making ongoing evaluation critical. So in summary, we're planning to evaluate the relationship between testing parameters and transmissibility of SARS-CoV-2 using viral culture as the gold standard. And this data will be supported with additional epidemiologic data from household contact transmission. And we hope this can allow for improved understanding of using diagnostic tests to assess for ability to transmit the virus. I just wanted to thank our PI, co-PI, Dr. Badley, Dr. Fida, and my mentors, Dr. Swift and Dr. Breer, and to our many labs that we're collaborating with on this research project for eventually getting this project off the ground. And thank you to the CERSI program for this opportunity.
Thank you. It's terrific. And obviously so critically important. I mean, anecdotally, I think we're all experiencing the sort of differences in transmissibility as people with new variants, obviously, but also I think the outstanding question, of course, is like, does it matter if in terms of vaccination status, in terms of transmissibility? It does not, at least with Omicron, does not appear to be.
Anecdotally, no, but we're hoping to get some answers here.
Yeah, it's terrific. Okay, well, I'll hold any other questions until the Q&A. Our next presentation is from Jaime Felser, who's going to be talking about disparities in vaccine preventable respiratory illnesses by race, socioeconomic status, and rurality. So welcome, Jaime.
Hi, everyone. My name is Jamie Felser. And
oh my goodness, I'm so sorry.
Oh, it's okay.
I'm so embarrassed. I apologize.
It's okay. I'm a second year pulmonary critical care fellow at Mayo Clinic in Rochester, Minnesota. And like was stated, I'll be discussing disparities in vaccine preventable respiratory diseases. So I have no disclosures. So during our talk, I'll be discussing the prevalence of respiratory vaccinations in high-risk adult populations, revealing the difference in influenza and COVID vaccination rates, and also highlighting the disparities of lower respiratory vaccine coverage by geographic region. So to give you a little bit of background on why we chose this topic, influenza and pneumonia are still in the top 10 causes of death overall, and influenza causes a vast number of hospitalizations every year with over 50,000 deaths a year. The amount of vaccination has been improving, but still we are vastly below the targeted amount. Healthy People 2020 had a target of 70% vaccination coverage for the high-risk population of adults aged 19 to 64. But in Minnesota, our highest coverage for the past 10 years was 53%. And as we have seen with the COVID-19 pandemic, it's disproportionately affected many underserved communities. And we recently crossed over that 1 million death mark, unfortunately. So we sought to understand this critical knowledge gap of disparities in vaccination coverage for respiratory diseases based on age, race, sex, socioeconomic status, rurality, marital status, and educational status. So as many of you are aware, vaccine recommendations currently are for influenza for everyone over age 6 months of years old. They're supposed to receive an annual influenza vaccination. For COVID-19, for those over 5 years of age, the recommendation is either 1 shot of the Johnson & Johnson vaccination or 2 shots of either the Pfizer or Moderna series. For pneumococcal vaccine, up until the recent recommendations were just earlier this year, for immunocompromised people, which are those with end-stage renal disease, multiple types of immunodeficiencies, either acquired or congenital, HIV, organ transplant, or those with congenital or functional asplenia or sickle cell disease, they're supposed to receive the pneumococcal conjugate vaccine 13, as well as a pneumococcal polysaccharide vaccine, and then an additional one 5 years later. The immunocompetent population is supposed to receive PPSV 23, and those are people with alcoholism, chronic heart, liver, lung disease, smokers, or with diabetes mellitus. CSF leaks or cochlear implants are supposed to receive both PCV 13 as well as PPSV 23 one time. And these are all the recommendations before someone turns 65. At that point, the recommendations do change, but we're going to focus on the high-risk younger adult period. So vaccine hesitancy has become more of an issue over the past 10 years or so. And recently there's been issues with vaccine disinformation, especially in the most recent pandemic, but it's been strongly influenced by this anti-vaccine movement that has been gaining traction. And overall, it's people think that they maybe don't need the vaccine, that it's unnatural or unsafe, and then they tend to resist the recommended vaccine schedules, which unfortunately further compromises herd immunity for those that can't receive live vaccines. The most commonly cited reasons for not getting annual flu vaccines are time, ease of availability, and concern for side effects, or concern that people will get the flu. However, recommendations by healthcare providers are often a top cited reason for those that did receive vaccination. So the outcomes for our study, our primary outcome is going to be vaccine coverage stratified by age, race, sex, individual economic status, marital status, geographic region, and education. Our secondary outcomes are the time to uptake from the diagnosis when they received their high-risk diagnosis, the relationship of COVID vaccines to influenza and pneumococcal vaccines historically, and the time people spent under vaccinated after receiving their high-risk diagnosis. So we included 4 counties in southeastern Minnesota and of adults aged 19 to 64 with these high-risk diagnoses. They had to have that high-risk diagnosis and be of that age group between 2010 and 2021. So we use the Rochester Epidemiology Project, or the REP, which has been around since 1966 and covers 99.9% of the population in Olmsted County, which is where Mayo Clinic is located. But it also incorporates many other health systems in the area and has now been expanded to cover 27 regions in southern Minnesota and western Wisconsin. And one of the major benefits is that it provides this extensive longitudinal population-based health regardless of where people receive their care. For our socioeconomic status, we decided to use an individual level of socioeconomic status because area level proxies tend to underestimate socioeconomic status and health. So we thought that this would be more accurate. Houses is something that was developed at Mayo and is a validated socioeconomic status that's based on publicly available housing characteristics such as square footage, number of bedrooms, bathrooms, estimated home value, square footage, and then where it's zoned. And this has previously been used to study other vaccinations in the pediatric population, but hasn't been used in the adult population yet. So getting to our early results, our cohort from 2010 to 2021, we had 45,000 people. They were evenly split between men and women. The majority were part of the immunocompetent group, with the largest of that group made up by chronic lung diseases, which included COPD, asthma, bronchiectasis, and cystic fibrosis, followed by diabetes mellitus. The largest group in the immunocompromised group was hematopoietic malignancies, followed by nephrotic syndrome and those on dialysis. So getting to some of our factors that we are looking for disparities in, this table highlights that, and you can see here that the majority of our population was white, non-Hispanic, living in an urban setting. They were married and more often non-smokers. The largest group was classified in the quartile 1 of our socioeconomic status, with an even split between the other quartiles. The majority of people received some college, about 35% of people did, and we had 21% who only received a high school or GED level, another 17% that completed college, and 21% that completed postgraduate studies. So into the results a little bit more, 68% of people received at least one flu vaccine during the time that they were eligible, which means that during that 11-year period, they were of that age group and had the high-risk diagnosis. So 68% of people received at least one flu vaccine, and 48% received 2 or more flu vaccines. We had almost 60% of people that were fully vaccinated for COVID-19, and based on the CDC guidelines right now, fully vaccinated is that someone's received their first series, so either 1 shot of Johnson & Johnson or 2 of Pfizer and Moderna. Up to date is classified as receiving the first series plus 1 booster. And so only 35% of people are up to date, meaning with the booster. Additionally, we found that there's positive correlation that was statistically significant between patients that were fully vaccinated for COVID-19 and received their influenza vaccine. And those that received COVID-19 vaccines were 39% more likely to receive an influenza vaccine. This is a chart of over the 11-year period of influenza vaccinations. And so the bar graphs are the number of people that received an influenza vaccine that year. And as you can see, the overall amount has continued to increase. The percentage of the eligible population that received the vaccine. So even though the overall number increased from about 2016 to 2019, we did have a decrease in the overall percentage, but it did increase last year. So looking at our logistic, this is a Poisson regression model of our influenza rate. We found that actually almost all of the variables were significant. The ones that are highlighted in red were the ones that weren't significant. So we looked at both continuous and a dichotomous age variable. And if we split it into over 50 and under 50, those that were over 50 years old were 24% more likely to receive their influenza vaccine. Females were 20% more likely to receive their influenza vaccine, and immunocompromised patients were 13% more likely to be vaccinated for flu. Those of Black race were 30% less likely to be vaccinated for flu, and those that classified as other mixed races were 8% less likely to be vaccinated. And then comparing people to the lowest socioeconomic status, all the other higher socioeconomic status groups were more likely to be vaccinated. And those that lived outside of an urban setting were 10% less likely to be vaccinated. Those that were unmarried were less likely to be vaccinated. And those with a higher education were more likely to be vaccinated. So looking at our COVID data, this is the up-to-date, meaning those that did receive both their primary vaccine and their booster. This cohort was 31,000 because we included December 1st, 2020 to December 31st, 2021, basically starting when the vaccine became available. And so the significant findings are here. There are still many, but slightly fewer than with the influenza model. And so those of older age are more likely to be vaccinated by about 15%. Females were 10% more likely to be vaccinated. And those of Asian race were 30% better vaccinated. And then those who were Native American were 35% less likely to be vaccinated. And also significantly, those that were college or higher educated were 1.5 times more likely to be vaccinated than others. And those living in rural locations were 10% less likely to be vaccinated for COVID, although overall, there was no statistical significance for that group. And comparing some of our other cohorts that we've evaluated, geographic region was one of the most significant factors in obtaining vaccinations. And so it was interesting that for COVID, this didn't quite play as much of a role. So the next steps are to complete the full analysis of the pneumococcal vaccines, finish up comparing them to pneumococcal, flu, and COVID-19, and seeing where there is discordance and concordance and who's getting their vaccinations. Eventually, we need to figure out why is coverage low for respiratory vaccines, especially outside of urban areas, those with lower education and of Black race. And then we need to act on these findings to increase the vaccination rates in this high-risk population. So in conclusion, influenza vaccination was associated with multiple sociodemographic factors, as well as race and ethnicity. COVID-19 and influenza vaccination are strongly correlated, and race, sex, socioeconomic status, education, and gender are associated with COVID-19 vaccination. I'd like to thank a lot of people, including CERSI, for the opportunity to do this research. I had some other funders as well, and would like to really thank my mentors, and I'll stop there. Thank you.
Thank you, Jamie. That was a great presentation. Maybe I'll ask one question just before we move on to top of mind, which is I wonder if you could speak to whether you think your findings will differ if you were looking at the flu vaccines, uptake 5 years ago versus it looked like they went way up during the time of COVID. If there was some kind of a sort of a confluence of concern and people went and got vaccinated for both when they got the other.
Right. No, thank you for that question. I think it will, and the goal is that we will look at the historical influenza data to see if there is concordance of the people that were getting, flu vaccines over the past 10 years, and then if they got their COVID vaccine, because like you said, it really shot up in 2020. And my guess is that may be related to the pandemic, and that people were encouraged to get both their flu and COVID vaccines. It went up like 4% from the year prior. So hopefully we'll continue on the upward trend, but yeah, that is something that we plan on looking at. Thank you.
It's really such great work. Okay, so let's go to our fourth presentation of the session before we go back to the Q&A for all the panelists. And we have our fourth presentation is from Usmeta Udikari, and that presentation is titled In Vitro Oral Drug Absorption.
Hi everyone. Can you hear me clearly?
Yes.
Great. Okay. Thank you for that introduction. So I'm Ashmita Adhikari. I am a PhD in pharmaceutical sciences at University of Maryland, Baltimore. So actually last week I defended my thesis and I'm in the transition of moving on to the another opportunity right now. So for this project, we actually did this project with the FDA, collaboration with the FDA, and we are also now in the process of submitting some manuscripts related to this CERSI project. So the topic of my presentation is in vitro prediction of in vivo oral drug absorption. So starting off, biopharmaceutics basically links that information for drug product quality throughout the life cycle of that product so that we can provide that patient-centric quality standards product. So what we are trying to see is that if we can gain that information in vitro when we perform like different studies in lab, have that link that can provide that relationship with what happens in human, what happens in terms of the in vivo performance of different kinds of drug products. That is basically an important understanding in terms of biopharmaceutics. So what we seek here is a solution test that could provide that predictive insight of what would happen to that drug product in vivo. And that is something which is really important when we are trying to understand different drug products, when we are trying to understand different biopharmaceutic risk assessment of these drug products. So this is something we are looking into for this specific project. So I would like to mention that with that, when we are trying to look for models, we are always chasing for that perfect in vitro model that would provide that insight into what happens in vivo. So what happened was when we started many decades ago, we have been using this one compartment dissolution testing for different drug products to basically see how they perform. However, this specific one compartment dissolution testing, it is not able to provide that prediction of what happens in vivo, and it often fails to give us that information of what is the performance like of these drug products in vivo. So different modifications have been done. These modifications, basically what we are trying to do here is to capture that dynamic interplay of dissolution and also that permeation that happens in human. So we made some contribution from our lab itself. Many years back, we came up with this continuous dissolution K-coder system. So this system, it is able to provide that dynamic interplay with the presence of this permeation component of K-code2 monolayer. One limitation that was identified was that this specific permeation setup, it has that low absorptive area to volume ratio of 0.5 per centimeter, which was actually leading to low mass transfer experimental hours. So we are trying to look what are the other permeation models so that we can incorporate that into this dissolution permeation setup. The existing other models like dissolution permeation setup, in vitro dissolution absorption system, they also have similar limitations. So we are looking into this new module, hollow fiber membrane module. It has this really high surface area of 115 square centimeter as compared to 1 square centimeter in existing in vitro model, so that the specific module could provide that higher dissolution and permeation setup that could provide that advantage of permeation model in this kind of in vitro setup. So this module have been found to show promise of by other in the literature study for different amorphous solid dispersion and establish that merit in terms of not just in vitro for different drug products, but also to in vivo performance of different drug products to utilize the specific module in dissolution permeation setup, utilizing that in vitro modeling so that we can have that insight into what happens in vivo in terms of drug absorption. So with that, the overall goal of this project was actually to evaluate the in vitro dissolution permeation setup, utilizing that novel hollow fiber membrane module. We are trying to anticipate the biopharmaceutic risk of different oral drug products. The main objective here was to understand that hollow fiber membrane module system to kind, what does it lie in terms of the permeation model, and we are trying to characterize that. We are trying to develop that in vitro model and also compare what we will see in the lab in vitro compared in human for different drug products. So we are comparing permeation constants and also the absorption profile for different drug products across various species classes so that we can evaluate this specific hollow fiber membrane module for a dissolution permeation setup. The hypothesis here was that when we utilize this hollow fiber membrane module, which has a really high area to volume ratio, that could be potentially used as an approach to predict drug absorption for different drug products. And we could see if that is able to provide that predictions of a drug absorption in vivo based on in vitro studies. So just to give you an idea of what was actually utilized for this specific project, what we did was we performed experimental studies and also we did model predictions so that we can come up with the system development. What we did was we utilized 4 different products: metoprolol, lamotrazine, piroxicam, and ranitidine. And what we did was we obtained the membrane, which basically comprises that hollow fiber membrane module in a simple flat sheet like a paper. And then we perform like permeability studies on them to get an idea of what is the permeability of this hollow fiber membrane module. We also derived 2 different models, a mixing tank and complete radial mixing model, which are well-known flow models. So we derived these models, these equations for our system. Based on this, we predicted our permeation of the specific system. And finally, we also performed experimental studies utilizing this setup. We compared those predictions and observation, and finally compared that with in vivo parameters. The in vivo parameters were based on the human data. So we obtained this permeation constant from the human data, and to compare that with the in vitro studies from our dissolution hollow fiber system, we obtained the in vitro permeation constant utilizing this equation. So based on this comparison of in vitro and in vivo permeation, based on this development we did, we are looking into the merit this hollow fiber system, this hollow fiber membrane module would provide for the predictions of in vivo. So here I'm trying to give you a brief idea of what is the system like, because the system is fairly new in this area. So I'm trying to help you understand what is the system like. So here what we did was we incorporated that USB dissolution apparatus with the hollow fiber membrane module. So what we can do is we can introduce our drug product, solution, tablets, capsules, simple formulations into this donor chamber. If you follow this blue line, it goes into this peristaltic pump, which is then introduced into the hollow fiber membrane module. This hollow fiber membrane module, it has 36 inner tubes, which actually comprises that high surface area of 115 square centimeter. To provide that hydrodynamics, to provide that continuous flow, which basically happens in vivo, what we did was we also introduced that buffer outside of this inner tubes, which would basically mimic the hydrodynamics, which is in vivo. So if you look closely here, what happens ideally is the free drug, which is dissolved in this blue compartment here, it is actually permeated through the membrane into this buffer, and then it is collected in the receiver. So what we see in the receiver is basically what is the drug that would be basically absorbed in the body, what would be the drug that would basically be absorbed after this whole setup of dissolution permeation. There is also this recirculation factor to provide that additional dissolution and permeation. One important thing I would like to note is that the area to volume ratio this hollow fiber membrane module provides is about 1.15 per centimeter, which is also comparable to the human A by B ratio, which is reported in the range of 1.9 to 11 per centimeter. So this was the setup that we utilized. So we derived 2 equations, a mixing tank model and also the complete radial mixing model. We utilized that recirculation factor since we performed recirculation in our system. We are utilizing initial parameters of membrane permeability that we obtained from that simple flat sheet. And also for our complete radial mixing model, we are also incorporating that flow rate that was incorporated in the system. These models were derived from by utilizing different parameters, understanding these parameters, and we are utilizing these 2 equations here. So just a brief result from our specific study. What we did was we performed these studies, experimental studies in 2 different volumes, 100 ml and 900 ml. So when we look at this graph of drug permeation versus all of these prodrugs that were studied, if you look in this black bar graph, we can see that it is about 40 to 50% in 100 ml, percent permeation. So these numbers mean really high in terms of these kind of in vitro models. So I would like to mention that this permeation, this high permeation that was seen for this system was actually a really big advantage when you are thinking of these dissolution permeation setup, when you are thinking of performing different absorption studies for different drug products. When we compare that with the predictions we made from our mixing tank and complete radial mixing derived models, so if you look into this graph here, we can see that there is the close agreement between experiment and also the model predictions in different volumes. So based on this study, we were able to develop this model, we were able to characterize this model, and then we also saw that close agreement in between the predicted and the observed values. So we were how does it compare with the existing models? So we are comparing with the side-by-side diffusion cell and the dissolution Keiko2 cell. When you look into this graph here of permeation versus all these prodrugs, we saw that there was really high percent permeation of around 15 to 20% in just 60 minutes, which is when you compare that to the other existing model, they just saw 1% and 0.1%. So that advantage, which was also correlated with the area to volume ratio, which is seen in this table here with the hollow fiber system, it provides higher A/V ratio as compared to the other models. With that close agreement, we wanted to see what is the relevance like in terms of in vivo. So we compared the permeation constant with the in vitro permeation constants of all these 3 different existing in vitro models. The DHFM system with actually showed really close agreement with the in vivo permeation constant with just about 5 to 10 fold compared to other models, which basically saw hundreds and thousands of difference in between these permeation constant parameters. So based on this, we were able to establish that merit of dissolution hollow fiber system as an in vitro model to provide in vivo predictions. We are also interested to utilize our system to see if it is able to differentiate in between different products. So what we did here and what I'm trying to highlight is the difference this DHFM system was able to provide in between immediate release drug product and extended release drug product. So if you look at this fraction absorbed versus fraction dissolved data, we can see that it was able to differentiate that this immediate release was a permeation rate limited absorption as compared to the extended release, it was a dissolution rate limited permeation, dissolution rate limited absorption. So these DHFM system, it was able to differentiate in between these 2 products and also differentiate in between low biopharmaceutics drug product and high biopharmaceutic drug products. This table here, I would like to show, we did like this study with other drug products, immediate release drug products, lamotrigine, ranitidine, and piroxicam IR, and utilizing our in vitro DHFM system, utilizing the fraction absorbed versus fraction dissolved data, we were able to establish that this DHFM system actually was able to differentiate in between low biopharmaceutics product in vitro, and also it showed concordance to what was in vivo for this specific products. And when we compared that with the ER product, the extended release product, it was able to differentiate in between these 2 products. So with that, my conclusions for this specific project was that this specific dissolution HFM system was able to provide that high area to volume ratio that provided really high drug